Did you know there are 10 million species currently live on Earth? Yep, that's 1 followed by 7 zeros. The amount of biodiversity is honestly astonishing, but it can make way for tricky classification in organizations for algorithms comparing different species with one another. With each of these organisms, there's billions, sometimes even trillions of cells to parse through. I'm Karthik, a high schooler interested in the field of machine learning and computational biology. In this video, I'll be going over the NCBI BLAST interface and how it can be used to reduce the time complexity of those algorithms that I just talked about before. Specifically, I'll be looking at homologous sequences, or genetic sequences that have been changed through evolutionary processes, and how it has changed the beta-catenin repeat protein of the thale cress flower. Due to the easy functionality of the NCBI interface, I was able to discover local similarities between variants of species in a computationally inexpensive and fast way. I'll be doing a demonstration of the NCBI BLAST interface, and then be showing the results. I had some interesting findings, so stay tuned until the end. Without further ado, let's get started! Okay, here we are in the NCBI interface. As you can see, there's a lot of stuff to scroll through over here, but for our purposes, we'll just be looking at the BLAST algorithm, or the basic local alignment search tool. As you can see over here in popular resources, you can access the BLAST algorithm through here, but I'll be showing a more simplified approach just so you can use it for any single protein that you want to search for as well. So I already have the accession number of my protein already done. So the accession number is kind of how the protein is identified. So in my case, I'm doing the beta-catenin protein for the thale cress flower. So now I can just press the search button, and as you can see, the NCBI is really great because it's a really big genomic database of all this information and all of these genes that you can just access in one go. And over here, I'll just press the BLAST button. And there's a lot of information over here. Over here, you can see that there is a query subrange. So over here, we're just going to put the values 33 to 100. And this is essentially just to say that we're going to take the 33rd nucleotide to the 100th nucleotide of this protein. Even though there are approximately 1,800 nucleotides in the beta-catenin repeat protein, we'll just take this for simplifying our purposes and kind of reducing the time that it takes to actually run through this entire process. All of the parameters should remain the same. So now let's just press the BLAST algorithm. And it should take around 10 to 20 seconds to spin up. So just to kind of go over the accession number again, that's a really, really simplified way of kind of exploring any single protein that you want to get. So each protein and each species of itself has their own accession number that it's usually identifiable by. And the .1 is essentially just the version number. So it'll be .2 if it was the second version. So over here, if we just scroll through this interface, we can see that there's a lot of information. So we have the scientific name, like the Arabidopsis Thaliana, which is basically just the thale cress flower. We have our total score and our max score. So those are the scores that basically how much that it does actually identify with the other nucleotides, meaning that how much does it actually align with one another. And then we have this e-value over here. E-value is another way of just identifying whether there's a local similarity between the other. If it's a low e-value, it's really good. So 2 to the 10 times 10 to the negative 35 is a really, really good value. And over here, the really cool thing is that we can see it in these. So BLAST is basically just searching for local similarities between nucleotide proteins, right? We can see over here that we're finding different species other than the Arabidopsis Thaliana. So we're finding the Camelina sativa, the Capsella rubella, and these are all different types of organisms. And it's kind of crazy how the NCBI interface can just scroll through this entire beta-catenin repeat protein, just go through each of the organisms and find these local similarities between over billions and trillions of proteins. So over here, if you just go to alignments, we can see how much of these alignments are actually with each other. So in this case, because it is the Arabidopsis Thaliana, which is what we were researching before, it will essentially have the same exact nucleotide sequence, so the same matchup as before. And this is also common over here and here. And as you can see, this is a really, really interesting finding because it reduces the computational runtime by so much by providing this easy and accessible database to kind of just store all your information. There's one last thing I want to show you guys. So this taxonomy thing. So you can basically find where in the taxonomy actually the Arabidopsis Thaliana is, or the thale cress flower is, and basically find the evolutionary lineage of it. Because remember, we're finding homologous sequences, so the biological sequences that are shown through that are related through evolutionary processes, right? So we can kind of backtrack and see that we maybe have something called the Brassica rapa, and we can see if there are any sequences that are related to that. So in my research, I kind of was just scrolling through these Camelina sativa, the Brassica rapa, and just going over all these different things and seeing how they kind of relate to the evolutionary lineage. And I saw very similar correlations. I mainly stick to the ones with really low E values and the ones with really high max and total scores. And through here, it's so easy to access everything. So over here, you can see this is the exact same accession number. So now if we just scroll down and check other accession numbers that we really, really want to find, we can just go down there and check it out as well. So, and of course, the percent identification is just how identical it actually is to the protein itself. So yeah, that was a really quick overview of how to use the NCBI BLAST algorithm. Please feel free to put any comments in the thread, or just comment me through YouTube, and yeah, I hope to see you in the future. Bye.